To introduce Dr. Jasna Kovac. And Jasna is, I think you have a long title. So Lester and Veronica Casida Career Development Professor of Food Safety and an Associate Professor of Food Science. Jasna completed her PhD in biosciences with an emphasis in microbial biotechnology in Slovenia, followed by a postdoc in the food science department at Cornell, and then joined the faculty in the food science department at Penn State. Jasna is highly recognized for her research and teaching achievements. And just looking at the past 2 years, she has been awarded 7 major honors. And a few of those include an Early Career Award from the American Society for Microbiology, ASM, an Outstanding Teaching Award from Penn State, and an Early Achievement and Research Award from the College of Agricultural Sciences. Jasna has a very strong dedication to undergraduate and graduate training and has helped organize multiple workshops each semester for the Microbiome Center. She also helps to run an NSF REU program and recently helped develop a T32 training grant proposal, which focused on integrating biotechnology and microbiome sciences education. So we are just very lucky to have Jasna as an executive committee member for the One Health Microbiome Center and a general leader for our community. With that, please help me welcome Jasna. We also have a plaque for you.
Thank you so much. Thank you, Nicole, for this nice introduction, and thank you for the opportunity to present here. I think last time I presented to the Microbiome Center was in 2018, so a long time ago. So today, the title of my presentation is Microbial Guardians, how microbiota shaped the fate of Listeria monocytogenes in food processing environments. So my lab is broadly focused on microbial food safety, and we work in 2 areas. First is focused on genotype-phenotype research and for food safety risk assessment. And then the second area is focused on microbiome phenotype research to elucidate the role microbiota play in the survival as well as persistence of foodborne pathogens in food processing environments and in food. In this first area, we study Bacillus cereus group quite a lot. So doing strain-based characterization to inform exposure and risk assessment. We have also been studying bacilli more broadly to predict their pathogenic potential based on genomic data. And then in the microbiome area, we have been studying the effect of microbiota on the survival and culturability of Campylobacter jejuni on food. We have used microbiome and metagenomic data to predict contamination of agricultural waters with salmonella. And then this last area that I will talk about today is looking at the effect of microbiota on Listeria monocytogenes, survival, persistence, and antimicrobial tolerance in food processing environments. But before I get any further into this, I just want to ask how many of you have heard of boar's head outbreak? Okay, I see a lot of hands up. If you haven't heard about it, I would encourage you to look at recent news. It's been all over the news. So this has been the most recent outbreak of listeriosis linked to sliced deli meats. It started late July. It's still ongoing. The investigation is ongoing. And there had been a recall issued. So they had to recall 7 million tons of sliced meat because of poor control of microorganisms, including pathogens in food processing environments. So far, 57 people have gotten sick through this outbreak in 18 different states. And 9 people have died. And actually last night they reported a 10th person have died. And CDC expect that this number will rise because serious symptoms of listeriosis sometimes don't appear until 10 weeks after the exposure. So For those of you who don't know what Listeria is, it's a foodborne pathogen. It's gram-positive pathogen. And it causes a variety of symptoms, from typical foodborne illness symptoms to flu-like symptoms and meningitis-like symptoms. And the most susceptible populations for listeriosis are pregnant women and older adults, and then generally people with weakened immune systems. So pregnant women, if they are infected, that can lead to miscarriage and stillbirth. And adults more often, older adults more often get serious complications, as well as immunocompromised individuals. So Listeria is one of foodborne pathogens that's quite unique in that it can grow at low temperatures. So it's not like Salmonella, which is an enteric pathogen. It will not really grow. At low temperatures, Listeria can do that. So it's very difficult to control in food processing environments. So it's not surprising that between 2009 and 2020, CDC documented 80 outbreaks of listeriosis that have been linked to 800 disease cases, and 86% of people who fall ill were hospitalized, and 16% have died because of this infection. So these outbreaks have been linked to a variety of foods that just shows how challenging it is to control it in various food processing environments. So soft cheeses, ice cream, deli meats, as we've seen most recently, and then also sprouts and mushrooms and lettuce, as well as cantaloupe and caramel-coated apple. So apples and tree fruit, that wasn't a food vehicle that government agencies really considered as high risk until 2014, when Listeria contaminated these caramel-coated apples and sickened a lot of people. So at that time, the scrutiny of controlling Listeria in these types of food processing facilities has really increased. And when I arrived to Penn State in 2017, I became really interested in that. So that's how this area of research was born in my lab. So last year, for example, there was an outbreak of Listeria linked to tree fruit, peaches, nectarines, plums. So that has caused 11 cases and 1 death across 7 states. So just to give you an example of what happens when an outbreak is detected, in that case, FDA and CDC work with local health officials and start to investigate the cause of the outbreak. And that includes sampling of fruit itself, as well as the environments where food is produced. So in this case, FDA went to these packing facilities, swabbed the environments, and they found 2 positive samples. So Listeria monocytogenes was isolated from these samples, but not only that, when they whole genome sequenced it, they found that the genomes are highly similar to those that were isolated from humans, which provided some evidence that there might be, these environments might be a source of contamination. So if we think about where Listeria resides, in what kind of environment, those would be agricultural environment, wildlife, natural environment, and water. So knowing that, it's not that difficult to imagine that contamination would enter a food processing facility with raw fruit, vegetables, and also tools that are used for harvesting. But what happens in the facility then, how the contamination is managed becomes really important to prevent further contamination of food, as well as exposure of humans. So as Listeria enters these food processing environments with food or tools, there's also other microbiota that contaminate these fruits and tools usually. So some of that microbiota will survive in food processing facilities, as well as adapt and then perhaps long-term colonize these environments. And that then provides an opportunity for some kind of interaction with pathogens. So common areas where Listeria often colonizes these facilities include drains and floors, especially if there's crack in the floors, as well as harvesting equipment, such as harvesting crates in the case of So if you look at this crate, you see it's built out of wood, and wood is very, very difficult to clean. And then there's also other parts of equipment that are difficult to clean. They're not very sanitary. So here I'm showing a piece of a roller brush that's part of a conveyor that's transporting fruits through different processes. You can see here a bunch of leaves degraded, and those were supposed to be cleaned and removed every day, but that is very challenging in reality. So these sites then present a very good harborage sites for long-term persistence of Listeria. And then because it's exposed to sanitation or sanitizers on almost daily basis, that can also lead to adaptation to antimicrobials, as well as antimicrobial resistance to, for example, efflux. Here I'm showing a few surfaces made of materials that are commonly used in food processing facilities. So we see here stainless steel. If we look at it under the microscope, we see that it's relatively smooth. And then high density polyethylene becomes a little more rough. And polyester nylon, the material that the conveyor belts are made of, is even rougher. And we see these cracks here that present really good harborage sites for microorganisms. And then the last one is plywood. You can see there's a lot of nooks and crannies where Listeria and other microbes can attach and becomes very difficult to remove. So the efforts that are made in these facilities to control pathogens include cleaning and sanitizing. So the purpose of cleaning is to physically remove organic matter, because we know that organic matter neutralizes and activates sanitizers, and that reduces their potential to inactivate pathogens. And then sanitizing is intended to reduce microbial load and particularly eliminate pathogens. When these sanitizing compounds are tested, they are usually tested in media that don't have high organic load. They are tested on clean surfaces. So if we see that there's a high log reduction by a sanitizer, that doesn't mean this is actually going to happen in a real life setting. What I'm showing here is a recent study from Oregon State University, where these researchers took coupons made of conveyor belt and coupons made of plywood. And they inoculated them with Listeria and then looked at what reduction they achieved with different sanitizers. As you can see here, quaternary ammonium compound sanitizer, bleach, and peracetic acid for both types of surfaces. And what they were particularly interested in seeing is what is the difference in the reduction when only sanitizer is used versus cleaner and sanitizer. And as you can see here, the general trend shows that including a cleaning step really improves the reduction of Listeria. And that is true for all sanitizers and for both surfaces. So this study has been done in lab. It has been done in very controlled environment where it's easy to evenly and consistently apply these chemicals to achieve good results, good outcomes. But in a real facility, that might be a lot more challenging. So what I'm showing here is the general process in a tree fruit packing facility, specifically apple packing facility. And these are just images from a YouTube video. This is not a facility that we work with. So we see a wooden crate that transports apples into the facility. And then apples are dumped into a dump tank that is filled with water. And sometimes there's antimicrobials added to that to reduce the load. And then apples go through this process of transportation on brush conveyor belts, and they go through washing process, followed by a drying process with pen drying, and then lastly, application of wax. And after that, they go into the packing procedure. So we were really interested in better understanding how effective cleaning and sanitizing protocols are in these facilities. And we were lucky enough to have 3 facilities on board to collaborate with us during the pandemic, which was very challenging. But with the help of fabulous students, it was possible. So in these facilities, we collected samples before and after cleaning and sanitizing under this conveyor belt made of brushes. So we sampled for Listeria. So we were able to determine whether Listeria is present or not, quantities of Listeria species, not just Listeria monocytogenes, and then also what is the total aerobic mesophilic bacteria concentration. And then lastly, we wanted to characterize also the composition of microbiota through amplicon sequencing. So to develop these cleaning and sanitizing procedures, we worked with chemicals industry that is developing cleaners and sanitizers. So we received a lot of recommendations from them, as well as industry groups, and developed these 3 treatments that I will be referring to as T2, T3, and T4. And then T1 was the baseline treatment that was different in different facilities, and sometimes it changed from one year to the other. And this project was done in 2 years. And I really want to thank these students, former students, who have done all the work for this project. So Marisabel, she was a master's student in my lab. She's now in plant pathology. And then Laura Roland, she's an assistant professor of food safety at Cal Poly now. And then Priscilla, she's working for Pepsi. So these treatments included a chlorinated alkaline foam cleaner, followed by a dewaxer. If you remember that workflow, there's a wax spraying procedure. And that wax is really difficult to remove with regular cleaners. So a specific cleaner we refer to as dewaxer was developed by industry for this purpose. And then once the cleaning process was done, we applied a peroxyacetic acid sanitizer to reduce microbial load. And then T3 included the same cleaner and dewaxer, but with the addition of an acid cleaner. So why add acid cleaner? Sometimes we have large portions of equipment made of stainless steel, and there's a lot of mineral buildup that can occur in these. So acid cleaner would dissolve that mineral buildup. And then that's followed by a sanitizer. And T4 was developed with keeping in mind the need to control biofilms. So the cleaners were the same, and that was followed by triple application of a biofilm remover. And that could help, or we hoped it would help in disrupting biofilms. And then once biofilms are disrupted, the sanitizer would inactivate Listeria and Microbiota. So what we found is that all of these 3 treatments resulted in a significant reduction of aerobic mesophilic bacteria, and that happened in both years. And this reduction was from 1 to 2.5 log 10 per swabbed surface, which means that about 10 to the 6 to 10 to the 9 cells per swab area survived through the cleaning and sanitizing. And then we looked at Listeria species population. And here we saw again significant reduction for all 3 treatments in year 1, but only treatment 4 resulted in significant reduction in year 2. So we suspect that maybe one of the factors that influence that might be the fact that in the 2nd year, we did not supervise the application of these treatments, whereas in 1st year we did. So here, generally, we saw 1.3 to 1.6 log reduction in Listeria species. And again, 10 to the 4th to 10 to the 5th cells per that swab area survived the sanitization. And then lastly, we want to know what happened with Listeria. So how frequently do we detect Listeria before and after application of these treatments? So generally, we saw the reduction for all 3 treatments in both years But the reduction was insignificant because we were underpowered in this study. We should have collected more samples. What was interesting to observe, though, is that about 2/3 of isolates of Listeria were collected in the waxing area, which suggests that that is really a problematic area that requires more attention in terms of controlling Listeria. So now we have a lot of isolates of Listeria, and so Marizabal went on to go genome sequence these isolates and then compare them with genomes of isolates that have been isolated in our previous studies, specifically in 2016 and 2017, when Luc Laborde's lab was doing a lot of research in these same facilities. So the purpose of this comparative genomic analysis was to see whether some isolates persisted over a long period of time, because our sanitation project was happening between 2020 and 2022. So once Marizabal had these genomes, she first genotyped them using multi-locus sequence typing to see if there's any specific sequence types that occurred in early years and later years. So she did identify some, and then she proceeded with single nucleotide polymorphism analysis and maximum likelihood tree construction. And these trees were mapped with spatial and temporal metadata. And again, the purpose of this was to identify whether any strains have persisted over a long period of time. So she found that sequence type 1003 and sequence type 445 were found both in early years and later years. And if we look at sequence type 1003, we see that all of these isolates that differ by just 2 to 4 SNPs were isolated from facility 2 from different areas, but that also included the waxing area. And then if we look at ST445, this one was also detected largely in facility 2 from different sites and over different years. You can see the color codes for years here. So in 2016, 2017, 2020, and 2021. So what's really interesting here and noteworthy is that in 2022, we no longer detected the genotypes. And we think that that might be because facility 2 has adapted our T2 on a daily basis and T4 on a weekly basis. So that might have helped with eliminating these persistent genotypes and controlling these facilities. And now hopefully the apples you buy in the store are safer.
Thanks.
So since these isolates were obtained from facilities where they are exposed to sanitizers, we wanted to better understand also their susceptibility to peracetic acid first here. And we found that majority of isolates were susceptible, their minimum inhibitory concentration was between 250 to 500 parts per million. If we compare that MIC with the application concentration, we see that it's above that. So this concentration wouldn't even inhibit Listeria in best case scenario. And it wouldn't kill it also. But then if we look at the biofilm remover, we see that the minimum inhibitory concentrations were far below the application concentration of 130. So we think that if anything was able to control Listeria in these environments, it was probably antimicrobial activity of this biofilm remover rather than peracetic acids. So because so much of that microbiota survived through cleaning and sanitizing, we wanted to better understand how these microbiota interact with Listeria. And the first step towards this understanding is seeing who's there. So we collected these samples and processed them for 16S rRNA amplicon sequencing to better understand the composition of microbiota in these facilities. So here we can see the general composition and how it differed among facilities. F1 is the purple one here, F3 is the yellow, and F2 is the pink one. So we see significant differences in the composition of microbiota in facilities. And for two of them, we also saw a significant difference between these two seasons where we conducted this research. But then when we look at the effect of the treatments, that was very inconsistent. For example, T2 worked well or significantly changed the composition of microbiota in facility 1 and 2 in year 2, T3 in facility 2 in year 2. So very inconsistent findings. And if we look at what microorganisms predominated in these environments, we see that in facility 1, Acinetobacter and Pseudomonas predominate. Same in facility 2. And then in facility 3, we mostly see Acinetobacter among most abundant bacteria. So now we wanted to better understand what is the relationship between the microbiota composition and Listeria contamination. So we conducted another project in these 3 facilities, and this one was conducted over 2 years or 2 seasons. And Ellie and Laura went to these facilities every 2 weeks and collected samples during processing. We again sampled under this conveyor belt and tested for Listeria monocytogenes culture and microbiota composition. What they found is quite alarming prevalence of Listeria. Here in F1, 28% of samples were positive for Listeria and 42 in year 2. In facility 2, all 189 samples were positive for Listeria. And then in facility 2, we saw a significant increase from 41% to 86% in Listeria prevalence. And again, a lot of these samples came from the vaccine area. That was actually what motivated us to do the sanitation project and help these processes. Next, we wanted to identify microorganisms that might be indicative of Listeria presence. So we did that by taking 2 approaches. The first one was differential abundance analysis, where we looked at whether there are certain ASVs that are significantly more abundant or less abundant in samples that are Listeria positive versus negative. So here we found Pseudomonas and Stenotrophomonas ASVs significantly more abundant in terms of relative abundance in Listeria positive samples. And our second approach was random forest. And here we found 30 different ASVs that were most informative for classification of samples into Listeria positive and negative. And those that were present in higher relative abundance included Pseudomonas, Microbacterium, and Flavibacterium. So some of our prior analyses were done at the family level. So we decided to try to isolate microorganisms, in general really, from these families from the packing environments. So Laura did this heroic work where she used this massively parallel isolation system that has arrays with over 6,000 wells. You can think of them as tiny chambers, growing chambers. That really helps grow microbes that are not fast growers, that are usually overgrown by fast growers like Pseudomonas. It gives them a chance to grow. And in these wells, you also have a metabolic dye. So if microbes are growing, that changes the color, and we can detect them with this machine spectrophotometrically and then further work with them. So Laura isolated thousands of these isolates and then randomly subselected 500, cultured them, extracted DNA, PCR amplified 16S rRNA, and then Sanger sequenced that and confirmed the genus. So that, not surprisingly, resulted in tons of Pseudomonas. But not all of her targets were isolated. So she continued with more targeted selective differential isolation to obtain other targets. So ultimately, she obtained Pseudomonadaceae, Microbacteriaceae, Xanthomonadaceae, and Flavobacteriaceae. And among Pseudomonaceae, she selected 8 Pseudomonas species for experiments. In Microbacteriaceae, she had 3 different Microbacterium species, Curtibacterium, Mulcibacterium, Agrococcus species. I haven't heard about many of these microorganisms before this project, so I can imagine all of you have. And then Xanthomonaceae, again, we have a lot of Stenotrophomonas here and Xanthomonas, and Flavobacteriaceae with different Flavobacterium species. So these were whole genome sequenced to identify them to the species level, but in a lot of cases, the genomes of type strains of these species didn't exist, or these microbes were just very new, so they haven't been taxonomically described yet. So one thing these microbes share in common is that they are psychrotolerant, So they can grow at low temperature that you would typically find in these facilities. And some of them are known to form biofilms like Pseudomonas. And we know that when microbes are growing in a biofilm where there's a lot of extracellular polymeric substances and a lot of repetition and everything, that they are more resilient to environmental stressors. And that really represents a challenge for processors for controlling them. So this is why it was important for us to understand which microbes co-occur with Listeria, and then how do they interact with Listeria, specifically in biofilms, and how do these biofilms affect the susceptibility or tolerance of Listeria to some. And that is important because it can improve the control of Listeria in these facilities. So what Laura did next is combine these microbes in different assemblages. So she had single, 2 family, 3 family, 4 family, and 5 family assemblages. And then she tested them for biofilm formation. So she had mixed family cocktails. They all contain Listeria, and she grew them in these MBEC plates. So these are essentially microtiter plates with a lid that has pegs on them. So it's just much better for biofilm testing than a 96-well plate because it reduces heat. And she moved in this nutrient-poor medium, R2A, 15 degrees for 3 days. Once biofilms were formed, she quantified the attached biomass using crystal violet assay, and then quantified Listeria monocytogenes using a probability number assay. And she also conducted some microscopy to really confirm and see how these biofilms. So what she found is that Listeria formed very little biofilm, as you can see here, compared to other assemblages. And most noteworthy was that increase in the biofilm that was formed was significant when Pseudomonas were present in the assemblage. So all of these included Pseudomonas. Then when she stained this with Cydo9 for live cells and tritium iodine for dead cells and look at them under a microscope, she also found that Listeria forms these very weak patchy biofilms. The same was true for Listeria with Microbacteriaceae and Listeria with Flavobacteriaceae. But whenever Pseudomonas and Xanthomonas or Xanthomonaceae were present, the biofilms were a lot more confluent, as you can see. And she also wanted to see whether concentration of Listeria increased in these biofilms. And she found that in all assemblages, Listeria concentration did increase. And this increase was lowest in the assemblage with Flavobacterium, which is consistent with that poor biofilm formation that we've seen in previous. And now to better understand which of these strains that we inoculated ended up persisting and showing up in final biofilms, she also did amplicon sequencing after PMA treatment. So to quantify only the viable microbiota in biofilm. And I'm not sure if you can see these ASVs that are mapped here. We were able to track ASVs because we selected microbiota to have at least 2 SNPs difference in V4 region. And we see that in Pseudomonas case here, there's always 3 ASVs that end up predominated in biofilms. So Sam, who's a postdoc in my lab, tested these individual isolates for biofilm formation and found that one of them is the highest biofilm former relative to others, but the other 2 are a little bit weaker biofilm formers. So it's possible that these have some other competitive advantage to end up persisting in the finding biofilms. Next, we characterize these Microbiota and Listeria for susceptibility to sanitizers. And we started with a quaternary ammonium sanitizer, benzalkonium fluoride. And we used a broth microdilution assay where lower MIC indicates more susceptible strains and higher MIC indicates less susceptible strains. And we found that Listeria had very low MICs. The highest was 6.25. And so relative to other microbiota, that was very low, especially if we look at these couple of Xanthomonadaceae and Pseudomonadaceae had much higher MICs. So now to study how these microbiota affect the tolerance of Listeria, which is not dependent on the concentration, but on the duration of exposure, we use double MIC of the most resistant Listeria isolate in the further experiments. Just to give you some background on tolerance, tolerant strains or population, they can have the same MIC as susceptible populations. But the difference is in the kinetics of the die-off, where susceptible population will die out faster, intolerant will die out slower. So the way we quantify this is by looking at the time of exposure that's required to achieve a 2-log reduction in population. And this is different than persistence, where in persistence, you have a small subpopulation in that low metabolic state or inactive state. And that can be seen by rapid die-off of the susceptible population and then this plateau in the persisters. And this is typically quantified by time required for a 3-log reduction in population. So our hypothesis was that Listeria monocytogenes will be more susceptible in a planktonic culture compared to biofilm culture. So we grew these biofilm assemblages again and exposed them to 12.5 ppm benzalkonium fluoride. And here in this graph, I'm showing the biofilm Listeria die-off in biofilms with a full line, and then Listeria die-off in planktonic form with these dashed lines. You can see in Listeria alone and Listeria with Flavobacteriaceae, the die-off in a biofilm is quite fast. It drops within half an hour, whereas planktonic population dies off slower. So this is not what we hypothesize. And we think it's because of that very weak biofilm where you don't really have a lot of biomass attached to these pegs. So the antimicrobial efficacy is greater than in a population, planktonic population where concentration is higher. But as we hypothesized, whenever we added Pseudomonadaceae, and as a matter of fact, any other family, we see this plateau in the die-off in a biofilm, whereas planktonic culture dies off faster. So we fitted Weibull model to this data to determine MDK 99, so minimum duration for killing 99% of the cells. And we found that that was over 2 hours, and sometimes it was infinity. It just wasn't possible to calculate based on the lack of die-off. And in contrast, we see from 20 minutes to 59 minutes for the planktonic culture. Now, 12.5 ppm is not a concentration that is applied in facility. It's much, much lower. So in reality, concentrations that are applied are around 200 ppm. So Olena and Laura then tested biofilms exposed to 200 ppm benzalkonium fluoride And they found rapid die-off of Listeria, Listeria with Flavobacteriaceae, and Listeria with Microbacteriaceae and Flavobacteriaceae. And in all other combinations, Listeria die-off was again attenuated, more or less. And then we also wanted to see what happens if we expose these biofilms to peracetic acid, specifically 250 ppm, which is commonly applied concentration. And here we saw much greater efficacy in inactivating Listeria in these biofilms. So these are all the assemblages where Listeria decline was quite fast. So it happened within less than half an hour. But every time we had Pseudomonas in these biofilm assemblages, the die-off was again attenuated, which just goes to show how important Pseudomonas is in achieving reduction of Listeria when there's biofilm formation. Then lastly, we worked with this biofilm removing agent. So we also wanted to see how effective it is in inactivating Listeria in biofilms, as well as total aerobic microbiota or all the other families. And we found a significant reduction in these populations in all tested combinations. And the reduction was over 4 logarithmic units. And here, when I have these dots, this is where the reduction was not below the limit of detection, whereas in other cases, it was below the limit of detection. So again, this is really valuable, practically valuable information for them because this chemical seems to be working very well in controlling biofilms as well as Listeria in biofilms. Now, with this, we gained a lot of applied insights into how to improve control of Listeria and biofilms. We know that if just peracetic acid sanitizer is applied, it has to be applied in a higher concentration, at least in this context. We know that biofilm remover is very effective in controlling not just biofilms, but Listeria in biofilms. And we also know that more attention needs to be paid to Pseudomonas in this environment, which is usually not, something processors think about. And while we did gain some ecological insights through these experiments, there's a lot more that we can do. So right now, Sam and McKenna are thinking about how to answer some of the following questions. How do Listeria monocytogenes and Pseudomonas interact? What is the role of interspecies as well as intraspecies form sensing in biofilm formation, stability, and behavior? And then also, how does Listeria monocytogenes respond to environmental changes that are caused by Pseudomonas? So there's definitely a lot more that needs to be done. And so with that, I would like to thank the funding agencies, the FDA, the USD, NEPA, that supported this research, as well as many people from my lab, Laura, Olena, Marisabel, Ellie, Hejong, Sam, McKenna, and then also Luc Laborde in his lab in our department, as well as our collaborators from FDA, Dimitri, Narjal. And thank you so much for your attention, and I would be happy to take any questions.
One talk. All right, we'll take some questions, and ideally, our first question will come from a trainee. Any trainees?
Hi, thank you. It was very interesting. I've been wondering that the first time you applied the low dosage of the aseptic of the clean material, what was the underlying assumption that how you chose exactly that dosage?
Yeah, so following this definition of tolerance that we used in this study, the MIC or the concentration of an antimicrobial should not influence the tolerance. So all we knew is that the concentration applied needs to be above MIC. So we decided to use double MIC of the most resistant Listeria monocytogenes isolate. So that was based on that assumption. But then the other experiment was done to make it more realistic.
Thanks, Jasna. I am not a food safety person, so I'm curious, like, what does the translation of these kinds of findings mean? Does this impact, like, potentially down the road FDA policy? Does this impact the way that commercial sellers are selling and telling people how to use these kinds of sanitizing treatments? Like, how does that work in the industry?
I think there's many steps before regulation is changed. So there's a lot of industry groups that are developing recommendations and guidelines for their industry partners. So working with them would be maybe one step towards disseminating this information, other than just publishing, because a lot of these people might not read scientific papers. So that, and then if we see that Pseudomonas is really problematic and kind of a red flag for in terms of food safety, maybe more studies are needed to see what is the rate of co-occurrence of Listeria and Pseudomonas, and thinking about what kind of evidence or data we need to collect to maybe consider Pseudomonas as an indicator of unhygienic conditions, because it's not used currently. It's different organisms that are used for this.
Thanks.
Was it difficult to get facilities to agree to let you sample? And then also, like, were there any, I don't know if repercussions is the right word, but like after you found all this Listeria and then you were like, hey, there's a lot of Listeria here.
So for me, it wasn't difficult because Luc Laborde did all the individual work, you know, so I kind of piggybacked on his relationships. So it was really great to have a senior faculty in the department when I joined that I could start collaborating with them. It was just amazing and continues to be. In terms of regulatory implications, all of our samples were collected from non-food contact surfaces, so they didn't have to be reported. But of course, if a facility knows they have positive samples in the environment, they need to make reasonable effort and corrective actions to eliminate that. And you know, the 100% prevalence was found in 2017, 2018. So we also felt like we have to do something to help them improve. So at least some of our research helped them improve a little.
Are the facilities required to have any kind of microbiological monitoring on a regular basis? And to what extent are there gaps that you anticipate a food microbiome laboratory should be more deeply involved in?
Yeah, so facilities do have to have environmental monitoring in place as part of their food safety plan. But regulators will not specify how does this look like. So you have a lot of freedom in identifying what are the hazards that you're trying to control, and you should be controlling based on the history of contamination and outbreaks. And then thinking about how are you going to control that and how frequently are you going to monitor and verify your critical control points. So if you're a small company and you don't have a food safety expertise there, this is really difficult. They don't know how to go about that. If you're a large multi-million company, you know exactly what you're doing. You have professionals that are hired to do that. So there's also that disparity in terms of access to resources. So that was also one of the reasons why we worked with these small packers.
What was it about the waxing that enriched for Listeria?
I think, and this is not based on scientific evidence, I think it's just wax layer kind of protects whatever microorganisms are on the surface. And if it's not removed with cleaning, maybe later it kind of opens up or peels off, and that releases microorganisms.
Yeah, so you showed that the Listeria would grow in the biofilm in the lab setting. But I'm wondering, in the packing houses, do you have a sense as to, is there a lot of contamination coming in that's persisting? Or is it a little bit of contamination that's actually replicating in leading to these high levels or relatively high levels?
We don't have the data to really be able to say whether it's one or the other. I think doing source tracking might help inform that, but we didn't sample orchard environments and tools, so I don't know whether it's one or the other. But just seeing these environments, 5 years ago, I think the conditions were such that they probably allowed for replication in the facility.
I want to follow up to Seth's question about vaccine. Because I feel like when I was thinking of apples, when they are get to us, we don't devax them. So I wonder, isn't that necessary to study when the wax is around? I know that you said that it will be easier to isolate or get to Listeria that way, but isn't that it would be, I guess, I don't know, is an unnecessary step to dewax it?
Okay, so we were looking just at an environment. So what's happening on the floor under that conveyor belt? So we never tested apples. And it is important to apply dewaxer in the environment because otherwise there's no way to clean these environments. And that then increases the risk for contamination with fruit. So I'm not sure if you were asking about dewaxing apples or the environment.
The apples.
Okay, yeah, that we didn't do. There's studies out there that also show how long Listeria survives, if it's in the wax or on the apple that's waxed.
This might be a little odd, but so thinking of human health, how we're kind of looking at things like phage therapy or fecal transplants. In a facility environment, are you getting any signatures of maybe beneficial microbes that are outcompeting Listeria or the ability to form biofilms? Or I'm sure phage therapy probably couldn't be applied there, but is there any beneficial hint of microbes that you could see?
Yeah, so I think microbes that we see, I guess microbes that are present more frequently when Listeria is absent could be potentially explored for this purpose. We have done some studies in the past looking at these fruit types. So specifically isolates that have previously been shown to be inhibitory towards Listeria monocytogenes. And we've seen that they work perfectly on a plate, 1-to-1 culture. They produce whatever they produce, like bacteria since. But when you add them to a complex microbiome and grow biofilms, all that benefit is diminished. So I think it's very challenging to identify and develop robust products. And I think this is similar for these cultures that are beneficial and used in soil and agriculture. It's just so much of the performance depends on the microbiome context.
So I'm thinking kind of like the upstream and where these microbes and Listeria come from, likely the field and the fruit. So I don't know if you've explored the data of maybe packing facilities that focus mainly on organic fruit versus conventionally managed fruit. Are there any differences in the Listeria load or anything like that?
In the facilities?
Yeah.
We haven't. I think there are some studies from the West Coast where apple production is much larger to look at that. I don't think there would be differences, honestly. I don't think that the practices that are used in the orchard would influence Listeria so much, but also there have been like large-scale genomic studies that have surveyed Listeria systematically across the United States. And they generally found that the prevalence of Listeria is lower in the West Coast than in the East Coast. And it has to do with soil composition and climate, or at least those factors were found as information.
Can you remind me, is all Listeria bad or are
It's
there
not.
and is any of your research able to identify like a bad one? More prevalent with certain
Yeah.
colleagues or?
So Listeria monocytogenes is a human pathogen. There are some that are animal pathogens, but they are not harmful to humans. And there's a lot of Listeria species. So in food science and food safety, we generally consider presence of any Listeria an indicator of conditions that would support the survival and growth of Listeria monocytogenes.
I guess, sorry, my question wasn't very well phrased. So if you find all these monocytogenes isolates, do any of them need to carry a particular virulence factor? or anything?
Yeah, so there's different kinds of Listeria monocytogenes. So there are generally 4 lineages, where lineage 3 and 4 are common in, natural environments, wildlife. And they are rarely seen in human infections. And then lineage 1 is very common in human infections, 2 is very common in food processing environments. So there's difference in pathogen.
All right, we're at 12 o'clock, so let's give another round of applause for Jasna.